Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biomolecules. So in the previous lecture we started with carbohydrates and then we saw we saw the structure of monosaccharides and disaccharides, and we also started studying about the polysaccharides. Among that, we studied about the structure of the starch. Now today we will continue with the more types of polysaccharides and we'll study their structures. So the first structure that we are going to study is about glycogen. So what is glycogen? The glycogen is the polymer of alpha D glucose. So glycogen is the polymer of alpha D glucose, and because it consists of only one type of monomer, it is homopolysaccharide because it has only one type of monomeric unit, that is alpha D glucose. Secondly, what are the linkages or glycosidic bonds present in the glycogen? So the linkages that are present in glycogen are alpha 1,4 and alpha 1,6 glycosidic bonds. So these are alpha 1,4 and alpha 1,6 glycosidic bonds. Now, if you remember, the starch that we studied in the last class also consists of this alpha D glucose as a monomeric unit, and it also have this alpha 1,4 and alpha 1,6 glycosidic bonds. So what's the difference between the starch and glycogen? So the first difference is that glycogen is more branched. It is more branched than starch. And because of this reason, it resembles amylopectin. It resembles amylopectin. So what is amylopectin? So in the starch, we study that it comprises of two components, the amylase and then the amylopectin. So this is the amylopectin was the branched part. So here the glycogen, because it is more branched, it resembles more of the amylopectin. If you look at the structure of the glycogen, so this is how a glycogen looks like. So here, this each unit is what? This is alpha D glucose, and they are linked together. So they have this linkage of alpha 1,4 glycosidic linkage, and where there is branching, the linkage is alpha 1,6 glycosidic linkage. Right. So now, what's the function of the glycogen? So the function of the glycogen, so it is present, glycogen is present only in animals. So the first thing is, you can write that these are present in animals. Okay. And the function is they have storage role. So they have storage role. They have storage role in animals. Okay. Now, so the structure I've just shown here, if I zoom in, this will look something like this, right? So this will be your glycosidic linkage and this will be your structure, right? And it will continue. The chain will continue on the both sides for n times, right? And then there will be also, at certain point, there will be branching that we can see. So here there is branching, and this branching will be where? Alpha 1,6 glycosidic bond. So the branching will be with alpha 1,6 glycosidic bond. So this is all about the glycogen. Now, the next polysaccharide that we'll study is inulin. So what is inulin? So again, inulin is a homopolysaccharide. It is also a homopolysaccharide. Why? Because the inulin, it is polymer of only one type of monomers, that is beta D fructose. So it is a polymer of beta D fructose. Okay. So because it has only one type of monomeric unit, it is known as homopolysaccharide. Now, what kind of linkage is present in inulin, or how this beta D fructose are linked to each other. So the linkage in inulin is, the linkage is beta 2,1 glycosidic linkage. So it's beta 2,1 glycosidic bond. Okay. So if you look at the structure, so here this is one fructose, this is second fructose. So these are what? These are beta D fructose. Now here the N is representing, this N is representing that there are N numbers of, or multiple, there are multiple fructose units, because it's a polysaccharide, and we know that polysaccharide consists of 10 to 100 monomeric units, right? So it has multiple fructose units. The one important thing is that at the end, or the terminal residue in the case of inulin, is the terminal residue, or the last residue is what? It's glucose. So the terminal residue is glucose, and that is alpha D glucose. Okay. So in the case of inulin, the terminal or last sugar is alpha D glucose. It is alpha D glucose. Now, where these are present? So the inulins are present in, these are present in tubers. These are present in tubers and roots. These are present in tubers and roots of a fruit known as artichoke. Okay. So inulins are present in the roots and tubers of artichoke. Apart from these, the inulins are also present in human. And what is their function, or why they are used? So in human, these are used for, inulins are used for testing kidney function. Okay. So in human, these are used for testing kidney function. Okay. And how the kidney function is being tested with using inulin? So here it is tested by looking the glomerulus filtration rate. By looking the glomerulus filtration rate, or you can say GFR. So by checking the GFR, the kidney functioning is being tested, and in this GFR, what is being tested? It's inulin. So this is about the inulin, that is the third polysaccharide, and it is also a storage polysaccharide. Okay. So inulin is also a storage polysaccharide, like glycogen and starch. So it's storage polysaccharide. Okay. Now, moving on to the next one. So the next one is the cellulose. So cellulose is, so till now we have studied about the storage polysaccharides. Now we are going to study about the structural polysaccharides. Okay. So cellulose is structural polysaccharide. It is structural polysaccharide, meaning it's structural polysaccharide, which means that it has role in structural support, role in providing structural support, right? Structural support. Now, it is also a homopolysaccharide, or you can say homopolymer. It is also homopolymer of beta D glucose. Okay. So it is a homopolymer of beta D glucose. So the glycogen and the starch are the polymer of alpha D glucose, while this is a polymer of beta D glucose. Now, what linkage is present in cellulose? So the linkage that is present in cellulose is beta 1,4 glycosidic linkage. So it's beta 1,4 glycosidic linkage. Okay. So it's beta 1,4 glycosidic linkage. So if you look at the structure, so this is your beta glucose, right? Because this is beta glucose because your OH is upside, right? That's why it's a beta D glucose. So this one is your beta D glucose. Again, this is also a beta D glucose. And then this beta D glucose, they'll link where? Because this is your position 1. And when you count, this is your position 1, 2, 3, 4, right? 5, 5 and 6. So the linkage is between 1, 1 and this one is 4th. This one is 4th. So this linkage is what? Beta 1,4 linkage. And here this N is representing that there are multiple number of this beta D glucose that are linked where in the same similar pattern, right? So this is how a cellulose structure looks like. And this is what? This is linear. Cellulose is linear. It is linear structure, right? The starch and glycogen that was branched, but this is the linear structure. Now, the important thing is that this cellulose, it cannot be digested by enzymes, human enzymes, okay? So the cellulose, the enzymes that we produce, it cannot be digested by those enzymes. So cellulose cannot be digested by human enzymes like amylase. So if you remember, the amylase is used to digest starch, right? And the only difference between the starch and the cellulose is the starch is made up of alpha D glucose, while the cellulose is made up of beta D glucose. Still, none of the human enzymes, including amylase, can digest the cellulose. So, what's the reason? Because the glucose, if you see, the structure is almost similar, except for the fact that at one position, the OH is located upward, that is in case of beta D glucose, while in the another case, it's alpha D glucose, that is in the starch, and the OH is located downward. So what makes this difference? That this cellulose cannot be digested by the human enzymes, while the starch can. So the difference is because of this beta 1,4 glycosidic linkage. So why it cannot be digested? So cellulose cannot be digested. Cellulose can not be digested by human enzymes because of beta 1,4 linkage. So this type of linkage that is making such a big difference. If it's alpha 1,4 linkage, it can be digested by human enzymes. But if it is beta 1,4 linkage, it cannot be digested. Okay. Now the question is then how it can be digested. So this is digested by another enzyme that is not present in the human. So digested by cellulases. So cellulose is digested by cellulases. Okay. But this enzyme is not present in the human. Now, where cellulose are present? So it is a major component of the plant cell wall. So when we were studying about the cell wall, we saw that cellulose is the major component of plant cell wall. Okay, it is a major component of plant cell wall. And apart from this, it is also present in cotton, wool, pulp. Okay. So this is about the cellulose, and we saw what is the difference between the cellulose and starch and why cellulose cannot be digested by human enzymes and starch can be. Okay. Now next we'll study about another structural polysaccharide, that is chitin. So chitin is structural. It is also a structural polysaccharide. Okay. It is also a structural polysaccharide. Now, it is the homopolysaccharide because again, it consists of only one kind of monomeric unit. Hence, it is a homopolysaccharide of N-acetyl glucosamine. N-acetyl glucosamine. Now, the question is, what is N-acetyl glucosamine? So it is also a kind of sugar moiety. Okay. So It is derived from glucose. So firstly, let's draw the structure of glucose. So how does glucose looks like? So the glucose looks something like this, right? It has a CS2OH, and then it's from the beta N-acetyl glucosamine. So OH will be here, H. Okay. So this will be your OH, this will be your OH, this will be your OH, right? And this is the structure of glucose, right? Now, in this glucose, if you remove this OH, okay, if you remove this OH and replace it with another group, that is NHCOCS3, then this becomes your N-acetyl glucosamine. So because this is first position, this is second, third, fourth, fifth, and sixth. So at second position, if you remove or replace the OH via this NHCOCS3, then it becomes N-acetyl glucosamine, or it is also called as NAG. Okay, so this is the structure of N-acetyl glucosamine that is derived from glucose. Now, what kind of linkage is present in this chitin structure? So it is beta 1,4 linkage. So it consists of beta 1,4 linkage. Okay. And so if you look at the structure of the chitin, so this is the typical structure. Here, each unit is what? NAG. So this is NAG, this is NAG. And then they are linked via this, you can see. So this is your first position, this is your second position. Similarly here, this is your first, second, third, fourth, fifth, and sixth. Sorry, the sixth position. So here, what you are seeing, that first position and fourth positions OH are participating in the linkage, and hence this linkage is what? Beta 1,4 linkage, right? Beta 1,4 linkage. And here this N is representing that there will be multiple units of this on the both sides. Okay. So this consists of beta 1,4 linkage, and this is a structure that is what? Linear. Again, chitin is of linear structure. Now, where this chitin is present? So chitin is present, it is present in the cell wall. It is present in the cell wall of fungi. So it is present in this, it is a component of the cell wall of the fungi. And apart from fungi, it is also present in the arthropods. Okay. So the outer covering of arthropods, that is mostly comprised of insects, right? So it is present in the outer covering of arthropods. So this chitin in the arthropods, chitin plus calcium carbonate, it makes the outer covering hard. Okay, it makes this outer covering hard. Okay, so this is about the chitin. Now, the next polysaccharide that we are going to study is peptidoglycans. Now, what are peptidoglycans? So peptidoglycans, these are heteropolysaccharides. Okay, so these are heteropolysaccharides. The first point is, till now we have studied about homopolysaccharides because they consist of only one type of monomeric units. But peptidoglycans are what? These are heteropolysaccharides, that they consist of more than one type of monomeric unit. Now, what is the monomeric unit of peptidoglycans? So the peptidoglycan, it is a polymer of N-acetyl glucosamine that we saw in the previous slide. So it is a polymer of N-acetyl glucosamine, and the second component is N-acetyl muramic acid. The second component is N-acetyl muramic acid. So we saw N-acetyl glucosamine, this is called as NAG, right? And N-acetyl muramic acid, it is called as NAM. So it is a polymer of N-acetyl glucosamine and N-acetyl muramic acid. Now, we have seen the structure of N-acetyl glucosamine. Now let's see how does N-acetyl muramic acid is, how it looks. Okay. So again, first let's draw. It is derived from, or you can say, extension of N-acetyl glucosamine. So let's draw first N-acetyl glucosamine. So what was that there in the N-acetyl glucosamine? So it has NHCO CS3 at second position, right? So this is your first position, this is your second, this is your third, fourth, fifth, and here it's sixth position, right? So here at second position, we have NHCOCH3 that makes it N-acetyl glucosamine. Now, in order to make N-acetyl muramic acid, what you'll have to do here, we have O, right? So this O, when it is linked by this muramic acid, then it becomes N-acetyl muramic acid, and the rest will be same, right? The rest will be same. So this is your N-acetyl muramic acid. So first you have to draw N-acetyl glucosamine by replacing the OH of the glucose at second position with NHCOCH3, and then at the third position you have to replace the H with muramic acid, that is what? CH3CHCOOH, and that makes it N-acetyl muramic acid. Now, this peptidoglycan, we have seen previously also, this peptidoglycan, it's the component of, or major, you can say major component of bacterial cell wall. So peptidoglycans are major component of bacterial cell wall, right? Bacterial cell wall. And how they are present in the bacterial cell wall? This is the structure. So here, this is G, this is M. So G is representing what? It is representing NAG, or N-acetyl glucosamine. The M is representing NAM, that is N-acetyl muramic acid. Okay. And now this NAG and NAM, they are connected via this. And on the NAM, we have this chain of, this chain is what? This is chain. This chain is a chain of amino acids. Chain of amino acids. And this amino acids helps in what? Cross-linking. Okay. And this is how the bacterial cell wall structure looks like. This amino acid helps in cross-linking. Okay. This helps in cross-linking. So this is how the peptidoglycan structure looks like. It is a chain of NAM and NAG, and then it also comprises of the chain of amino acid in the bacterial cell wall that helps in the cross-linking. So this is about the peptidoglycan, and again, it is also what? It is structural polysaccharide. It is also structural polysaccharide. Why? Because it is present in the structure, right? It is present in the bacterial cell wall. The next structure that we are going to study is, apart from this, there are certain other sugars, you can say. For example, ascorbic acid. So ascorbic acid. Now this ascorbic acid, it is also derived from sugar. Okay. It is also derived from sugar. Okay. It is derived from sugar. Then we have certain other heteropolymers such as hemicellulose. So we need not to study this in detail, but just for the name, that what are the heteropolysaccharide example. So hemicellulose, hyaluronic acid. So these are all polysaccharides, hyaluronic acid. But the difference is that these are heteropolysaccharides. Okay. Chondroitin sulfate. So these are what? These are heteropolysaccharides. Okay. These are heteropolysaccharides. Now, apart from this polysaccharides, let's study the last polysaccharide, that is mucopolysaccharides. Okay. So what are mucopolysaccharides? So mucopolysaccharides are, these are the polymers of modified sugars. Okay. These are the polymers of modified sugars like NAG and NAM. Because NAG and NAM are what? They are also modified sugar, right? They are generated or produced by modifying the sugar such as glucose. Okay. So mucopolysaccharides are also the polymers of modified sugar like NAG, NAM, etc. Okay, NAG, NAM, etc. And if we talk about the example of mucopolysaccharides, so the examples are, the first example is glycocalyx. Okay, the first example is glycocalyx, and where the glycocalyx is present. So it is the outermost layer of bacterial cell wall, right? So the glycocalyx, it is the outermost layer of bacterial cell wall. Bacterial cell wall. Okay. Then apart from glycocalyx, we have also mucilage that is present onto the surface of cyanobacteria. Okay. So the outer mucilage in the case of cyanobacteria, in cyanobacteria. So these are the examples of mucopolysaccharides. Okay. So with this, we have completed another biomolecule, that is polysaccharide. So till now, we have completed what? We have completed proteins and carbohydrates. And in proteins, we saw amino acids, enzymes, and structure of proteins. And then we have also completed carbohydrates. Now, we are going to move to the next next biomolecule, and that is the next biomolecule, and that is lipids. Okay. So, what are lipids? So, let's see. We have studied this term. We have often heard this term that in the food it has a lot of lipid, or you can say a lot of fat. So now let's see what are lipids. So just like protein and carbohydrates, lipids are also organic compounds, right? Because these are biomolecules, and we saw the definition of biomolecules. Biomolecules are what? These are organic compounds. So lipids are also organic compounds. So these are organic compounds. And if these are organic compounds, then it contains, so these are organic compounds that contain what? Carbon, hydrogen, oxygen. So these are three mandatory things that will be present in an organic compound, right? But apart from these, based on the type, the lipids can also consist of what? Phosphorus, nitrogen, etc. Okay. So these are the main compounds, and these are the compounds or elements. Sorry, these are CHO are the main elements, and then based on the type, it can also consist of different other elements such as phosphorus, nitrogen, etc. Okay. Now, lipids. If we talk about the features of lipids, so the lipids are insoluble in water. Lipids are insoluble in water. Okay. Now they are insoluble in water. So in what the lipids can be dissolved? So lipids, these are soluble in, lipids are soluble in non-polar solvents. Okay. These are soluble in non-polar solvents. Okay. They are soluble in non-polar solvents. Now, if we talk about general formula of lipid, so the general formula of lipid is CN. Sorry. So the general formula will be CnH2O, just like what we saw in the case of carbohydrate. So it will CN H2N O2. So the only difference is here the number of oxygen molecule is fixed. Okay. So number of oxygen is fixed. It will be what? That is 2. Okay. So it is fixed. Apart from this, it has CnH2n. Okay. So that is, if there is n number of, or there is 5 number of carbon, then the hydrogen will be 10, but oxygen will be 2. Okay. Now, what the lipids are made up of? So lipids are made up of two components. The first one is glycerol, and the second is fatty acids. Okay. So lipids are made up of glycerol and fatty acids. And how we can digest or hydrolyze lipid? So lipids are hydrolyzed. These are hydrolyzed by enzyme called as lipases. Okay, so it is hydrolyzed by the enzyme that is called as lipases. So lipase, so when it will hydrolyze the lipid, the what components you will get? You will get glycerol and fatty acids. Okay, so this is the about the introduction of the lipid. Now, next thing that we are going to study is fat. So the people often gets confused between the term lipid and fat. And we always use these terms together that lipid is fat, fat is lipid. But is it really true? So fat, we can say, yeah, fat is lipid, but we cannot say that all lipids are fat. Okay. So what is fat? Fat is a subgroup of lipid. Okay. Fat is subgroup of lipid. Okay. Which means that all lipids are not fat. Okay. So fat is subgroup of lipid. Okay. So lipid is a broad, broad category. This is your lipid. And here somewhere you have a small category that is your fat. Okay. Now, then what will be the structure of the fat? So remember, I told that the lipid comprises of glycerol and fatty acids, right? So here you have glycerol structure that looks something like this: CS2OH, CHOH, and CS2OH, right? This is a fat. Sorry, glycerol. This is a what? Glycerol. Now, your fatty acids. You have fatty acid. We will see in the next slide how fatty acid look like. But if the structure forms something like that, this is fatty acid that I am representing by Fa. Okay, CS2O, sorry, CHOFA and CS2OFA, which means that all three H, this H is replaced by the fatty acid. Then this structure will become fat. Okay, this will be called as fat. So, what is fat? Fat is when all the three hydrogens, when all the three hydrogens of hydroxyl group is replaced by fatty acid, then it will be called as fat. Okay, so fat is when all the three H of the hydroxyl group is replaced by the fatty acid, then that will be called as fat. Okay, so the fat will look something like this. Now, what are the role or function of fat? Because we often say that we should not take a high fat diet, right? But is it like the fat is always bad? The answer is no. The fat plays variety of important roles in our body. Okay, so the first function is it provides energy. So this fat, it undergoes oxidation. Okay, and provide energy. Okay. Secondly, it keeps the body warm. So it acts as an insulator to heat. So it does not allow the heat from inside the body to go outside. Okay, so it acts as an insulator and hence it keeps the body warm. So thirdly, it protects the internal organs. So it also acts as a shock absorber. Okay, it also acts as a shock absorber, and by this way it protects the internal organs. Fourth, it also helps body to absorb vitamins. So there are different types of vitamins. So, but this four vitamins, that is ADEK, ADEK. So these are what? These are fat soluble vitamins. So these are fat soluble vitamins. Okay, these are fat soluble vitamins, and this fat helps in the absorption of these vitamins by the body. Okay, because the ultimate goal of whatever we eat is their absorption. Once they helps in the absorption of these vitamins, that is ADEK. Okay, next it also helps in the production of hormones that help body to work properly. Okay. So these hormones are what? These are known as steroids. These hormones are known as steroids. And then it also helps in act as a act or use for the storing substances for the body extra calories. Okay. So these are the various function of fat. Now we have seen what is lipid, what is fat, and we can also now understand the difference between the lipids and fats. Right. Now let's talk about the fatty acids. So we have seen the structure of the glycerol, but what is the structure of the fatty acid and how does it looks like? Okay. So the fatty acids, these are the long chain of hydrocarbons. Fatty acids are what? These are the long chain of hydrocarbons. Okay. These are long chain of hydrocarbons with a carboxylic group. With a carboxylic group. Okay. With a carboxylic group. Okay. So this is a long chain of hydrocarbon with a carboxylic group. So if you draw the structure, it will look like this. It will look like this. Okay. So at its one end, we have what? Carboxylic group. We have carboxylic group. And then we have this long chain of hydrocarbon. This is the long chain of hydrocarbon. Okay. Now, the fatty acids are of two types. So if we talk about the types of fatty acids, we have two types, the saturated fatty acid and unsaturated fatty acid. So what is the difference between saturated fatty acid and unsaturated fatty acid? So in the saturated fatty acid, there is no double bond. Okay. So in the saturated fatty acid, there is no double bond. Okay. So its structure will be like this. There is no double bond. Okay. But in the case of unsaturated fatty acid, it has double bond. Unsaturated fatty acid have double bond. Okay. So its structure will look like this. It has COH and then it will have double bond at various places. Okay. So this is how the unsaturated fatty acid will look. Now, this saturated fatty acid, it has high melting point. High melting point. Okay. It has high melting point, while the unsaturated has low melting point. It has low melting point. Okay. Next, because it has high melting point, so it is at room temperature, it is solid. So saturated fatty acid is solid at room temperature. So do we know any fat or we can say, do we know any lipid that is solid at room temperature? So have you seen butter, ghee? They are mostly solid at room temperature, right? So if we talk about the saturated fatty acid, the example can be a butter, ghee. Okay. So these are, and this butter, ghee, this mainly constitutes what? Fat. So comes under fat. Okay. So saturated fatty acid that you can say consist fat. Okay. While the unsaturated fatty acids, so they are liquid at room temperature. Liquid at room temperature. So we always see in our houses that there are lipids, certain lipids that are liquid at room temperature. And they are what? These are oils. So the unsaturated fatty acid, they mainly comprises of oils. So whatever oils we are talking about, these are unsaturated fatty acids. While the ghee, butter, these things that are solid at room temperature, these consist of what? Fat, or they comprises fat. Okay. Now, another difference between saturated and unsaturated is that the saturated fatty acids are originated from animal. So they have animal origin. The saturated fatty acids have animal origin, while the unsaturated fatty acids, they are originated from plants. Okay. These are originated from plants. Okay, these are originated from plants. Now, these are of animal origins, and where these are stored? So these are stored in the liver. In animal, these are stored in liver and beneath skin. Okay, so below the skin, they are stored in the liver and beneath skin, while in unsaturated fatty acids, because they are originated from plant, so they are stored in seeds and fruits. Okay, we get oils from where? From seeds, right? So these are stored in seeds and fruits. Okay, they are stored in seeds and fruits. Apart from this, there are certain other differences between unsaturated and saturated fatty acid. The next is the saturated fatty acids. These are stored in the animals. These are stored in the special cells. Okay, so these are stored in special cells. Okay, these are stored in special cells, and these cells are called as adipocytes. These are called as adipocytes. While the unsaturated fatty acids, these are stored as granules. These are stored as granules in plants. Okay, these are stored as granules in plants. Now, the saturated fatty acids, these are generally unhealthy. Okay, so they are generally unhealthy. These are generally unhealthy. Unhealthy means because they increase the blood cholesterol level. So they increase the cholesterol. Okay, they increase the cholesterol, and we know that high amount of cholesterol in the body is not good. Okay, because it increases the risk of cardiovascular diseases, right? So it often happens that people with high blood pressure or patient with high BP or high blood pressure are not recommended. So they are not recommended. They are not recommended to take saturated fatty acids. Okay. So they are not recommended to take saturated fatty acids, right? But if we talk about unsaturated fatty acids, so they are healthy. So these are generally healthy. Okay. These are generally healthy. And the reason is because they decrease cholesterol level. Okay. They decrease cholesterol level. Okay. Now, if we look at the examples of saturated fatty acids, okay. So there are various examples of saturated fatty acids based on the number of carbon molecules it has, based on number of carbon, molecules it has. But we have to just remember two. Okay. So the first is palmitic acid. Palmitic acid. Now, how many carbon molecules are present in the palmitic acid? So it has 16 carbons. Okay. And we also know the general formula of lipid, right? So if someone gives you the palmitic acid, it has 16 carbon. So you can write the formula, right? The chemical formula of palmitic acid. What will be the chemical formula of palmitic acid? It has 16 carbon, so it will be C16. And then what we have? We have what? Cn, S2n, O2, right? So we have C16, H2n means 32, and O2. So this is the general formula or the chemical formula of palmitic acid. So one example of saturated fatty acid is palmitic acid. The second is stearic acid. And how many carbon moieties have stearic acid? It has 18 carbons. Okay. Now, apart from this, these two are important to remember. Then apart from this, we also have arachidic acid. Okay. Arachidic acid. Arachidic acid, it consists of 20 carbons. And then we have lauric acid. And it comprises of 12 carbons. So these are certain examples of saturated fatty acids, but the two palmitic acid and stearic acid is important, and you should remember how many carbon molecules are present in it. And because it is saturated fatty acid, it won't have any double bond. So we can draw the structure of both of these. We have to make a hydrocarbon chain of 15, and then we have one COH group. Okay. So this 16 carbon is including the COH group, carboxylic group. If someone asks you that how many carbon molecules are present excluding the COH group in the palmitic acid, then the answer will be 15, right? Because this 16, it includes the carboxylic group. Okay. Now, moving to the unsaturated fatty acids. So the unsaturated fatty acids, these are of two types. So the unsaturated fatty acids are of two types. And what are the two types? So these unsaturated fatty acids, these are of two types based on the number of double bonds. Okay, based on the number of double bonds. So the first type is monounsaturated fatty acid. So what is monounsaturated fatty acid? This is also known as MUFA. Okay, this is also known as MUFA, monounsaturated fatty acid. Okay. Now, as the name suggests, mono, it means single. Okay, so it contains single double bond, right? Or only one single or only one double bond. Okay, it consists single or only one double bond. Okay, and what is the example of monounsaturated fatty acid? The example is oleic acid. Oleic acid. Oleic acid, it has 18 carbon. It has 18 carbon and one double bond, right? So it is monounsaturated fatty acid. Now the second type is polyunsaturated fatty acids. So what are polyunsaturated fatty acids? These are also called as PUFA. Okay, these are also called as PUFA, that is polyunsaturated fatty acids, and it contains more than one double bond. Okay, it contains more than one double bond. Okay, it contains more than one double bond. And the three examples that are important are linolenic acid, this is L, linolenic acid, then linoic acid. Just a second. I'll write in the order so that it will be easy to remember. Okay. So the first is linoic acid. Okay. The first is linoic acid. Second is linolenic acid. Linolenic acid. And the third is arachidonic acid. Arachidonic acid. Okay. So sorry, it's linolic. It's linolic acid. Okay, linolic, linolenic, and arachidonic acid. Okay, the first one is linolic acid. Okay, so it's linolic acid. Okay, linolic acid, linolenic acid, and arachidonic acid. Okay, so the linolenic acid, how many double bonds it has? It has 2 double bonds. Linolenic has 3 double bonds and arachidonic has 4 double bonds. So 2, 3, 4 is its number of double bonds. Okay, its number of double bonds. So this linolenic and linolenic, it is of 18 carbon. Okay, so these two are of 18 carbon, while the arachidonic is of 20 carbon. Okay, it is of 20 carbon, right? So this is the example of polyunsaturated fatty acid. The one important thing is that more the number of double bonds or more the degree of unsaturation. So more the number of double bonds. Double bonds means higher the degree of unsaturation, right? So the more the number of double bonds, healthier the fatty acid. Okay, healthier the, you can say, lipid. Healthier the lipid. So it's good, or it will be better to take the polyunsaturated fatty acids that have high number of double bond. For example, arachidonic acid that is present in the almond. Okay. So more the number of double bonds, healthier the lipid. Now, if someone asks you the example of monounsaturated fatty acid, you can also write the mnemonics for this is OLA, OLLA, where this constitutes PUFA, that is your linoleic acid, linolenic acid, and arachidonic acid. And this is your MUFA, that is monounsaturated fatty acid, and it is stands for oleic acid, right? So this is about the unsaturated fatty acids and their types. So we have seen the fatty acid and their type. Now next, let's move to the classification of lipids. So the lipids are classified into three types. Lipids are classified into three types, and these are the first one is simple lipid. The first type is simple lipid. Okay. The second type is called as conjugated or compound lipid. Conjugated or compound lipid. And the third is called as derived lipid. Okay. Third is derived lipid. So the example of simple lipid is fat. Okay. Wax, cuteins, or suberines. Okay. Or suberines. So these are the example of simple lipid. The conjugated or compound lipids, example are phospholipids. These are phospholipids. So conjugated means they are attached or linked with a certain group, right? So phospholipids, glycolipids, lipoproteins. These are the example of conjugated lipids. And derived lipids, examples are sterols, prostaglandins. So these are the example of derived lipids. Now we will study about these lipids one by one. Okay. So the first type of lipid is simple lipid. Okay. So what does simple lipid consist of? So it consists of glycerol. It consists of glycerol plus fatty acids. Okay. It comprises of glycerol plus fatty acids. Okay. So how does simple lipid is formed? So we know the structure of glycerol. It's CS2OH, CHOH, and CS2OH, right? CS2OH. Now, suppose here comes a lipid. Okay. So the lipid, let me draw like this, HOOC. So this is your COH group. And then there is long chain of hydrocarbon. Okay. Now what happens? What happens is this OH and this H, it forms a bond and release. They release a water molecule, right? And this leads to the formation of a bond. Okay. This leads to the formation of bond, and it will look something like this. CS2O, CO, and then you have long chain of hydrocarbon, right? And then here you have CHOH and CS2OH. Now, what this bond is called as? This CO, what this bond is called as? This bond is called as ester bond. Bond is ester bond. So in lipids, what kind of bond do we have? We have ester bonds. Okay. And the lipases, what bond they break? Because we saw in the first slide of lipid that lipids are digested or hydrolyzed by lipases. So this lipases breaks this ester bond. Okay. Now, the simple lipids are of three types based on the number of fatty acids it has. Okay. So the simple lipids, these are of three types based on the number of fatty acids it has. The first type is known as monoglycerides. The first type is known as monoglycerides. Now, what do we have monoglyceride? It contains one fatty acid. Okay, it contains only one fatty acid. Okay, it contains only one fatty acid. So it means that it is made up of what? It is made up of one glycerol, right? It is made up of one glycerol and one fatty acid. Fatty acid I am representing by F. Okay. And if we look at the structure will look like either this or what I draw or like this. Here you have first, second, third. This is your glycerol. And then this part is your fatty acid. Here this R is representing the chain of hydrocarbon. Okay. The R is representing the chain of hydrocarbon. So this is how the monoglycerides look like. Now, the second type is diglyceride. Second is what? Second is diglyceride. So what is diglycerides? As the name suggests, it has two fatty acids. Okay. So diglyceride, it comprises of one glycerol. So the glycerol number will be same. One glycerol plus two fatty acids. Okay. And the structure, if you look at, the structure will look like this. So this is your glycerol. This is your glycerol. And the fatty acids, this is your first fatty acid and this is your second fatty acid. Again, this R1 and R2, the R1 and R2, they are representing chain of hydrocarbon. Right? They are representing chain of hydrocarbons. Okay. So this is basically the red one is what? Your fatty acid, and this black one is glycerol. Now the third type is triglyceride. The third type is triglycerides. Now what are triglycerides? So the triglycerides, it consists of one glycerol. Again, the glycerol number is same, right? So it comprises of one glycerol plus three fatty acids. It comprises of one glycerol plus three fatty acids, and the structure looks something like this. So again, this is your glycerol. The black one represents the glycerol, and the red one represents the fatty acids. It is representing fatty acids. Now, this glycerol or this structure is known as true fat, or the fat that I was talking about. So, if someone asks that what are fat? So, fats are what? These are triglycerides, because in this case, each of this H of the OH group of glycerol is substituted by fatty acid. Okay, and this is called as true fat. And what are fats? Fats are triglycerides. Fats are triglycerides. Right? Fats are triglycerides. So this is about the simple lipids. Okay. Now the next that we'll study is conjugated lipid, or we can also say as compound lipids. Okay. So what are conjugated lipids? Conjugated lipids are fatty acids. These are comprises of fatty acids plus glycerol plus an additional group. Okay. So it also comprises of additional group. So this is a conjugated lipid. Okay. Now, the examples of conjugated lipid we saw. So the first example is phospholipids. Example of conjugated lipids are, the first example is phospholipids. Now, phospholipids, these are present where? So, phospholipids are present in cell membrane, right? Phospholipids are present in cell membrane. So, the majority or the major component of a cell membrane is phospholipid. Now, what is phospholipids? Okay, if you look at the structure, if you talk about the structure, what are phospholipids? So the phospholipid, it comprises of diglyceride. It comprises of diglyceride plus phosphate. The additional group that I was talking about is phosphate. Okay, so let's draw the structure. So we just in the previous slide we saw that how does a diglyceride look like. So the diglyceride have CS2O, CO, and R1 plus CHO, CO, R2, right? And then the third will be your CH2, CS2O. Okay, here H. but here you will have H. So this makes your diglyceride. Sorry, it makes your diglyceride, right? It will make your diglyceride. Now, what else? Next thing, what it is saying that you have diglyceride plus phosphate. So this phosphate, it will come, okay, it will be like this, okay, this phosphate. Now this phosphate will form a bond. Here also you have OH, and this phosphate will form a bond by release of water molecule. So in biomolecules, if anything is formed, the water molecule is released. Okay, so a water molecule is released, and the structure will become what? The structure will become. Just a second. So the structure will become something like this, or I can draw it directly here. So the structure will become like this. Okay, so the structure, it will form a bond directly here, and it will become like this, right? It will become like this, and this structure will become your now phospholipid. It will become your phospholipid. So we saw that how this phospholipid is formed, because at the third position, this is a 1, 2, 3. At third carbon, the H is substituted by this phosphate group, and hence it forms the phospholipid. Now, when we were studying about cell membrane, we saw that the lipid looks something like this. So we represented lipid with this, right? So this circle, what we called it as head. And this head is nothing but this region. Okay, this region. Till here, it is head. And then this is called as tail. This is called as tail, which is represented by this region. Okay. And this head was what? It was hydrophobic. The head was hydrophobic, while the tail was hydrophilic. So it was located in the cell membrane, it was located outside, while the head was located inside. Okay. So this is how the phospholipid structure looks like. Now, apart from this, we have one more example. We have one more example, or you can say type of phospholipid, and that is lecithin. Okay, that is lecithin. So where the lecithin is present, it is present in cell membrane. It is present in cell membrane, plus it is also present in alveoli. It is also present in alveoli. Okay, and what it consists of? It consists of fatty acid plus glycerol plus one more group that is called as choline. Okay, and what is the structure of choline? So the choline, it looks something like this: CH2, and CH3, CH3, CH3. Okay, so in this case, instead of all the structure will be same, it will have fatty acid, glycerol, and one more component it will have phosphate. Okay, so if you want to draw the structure of choline, you have to draw the structure of phospholipid, and in this phospholipid, with this phosphate group, you have to also add this choline. Okay, so I can quickly draw the structure of lecithin. So the lecithin structure will look something like this. You have your CH2O CO R1. This is your CHO CO R2. Then you have your CH2O. This is your phosphate OH. And this O, it will form a bond with the choline, that is CH2, and then you have CH3, and you have CH3, right? So this is your structure of lecithin. Okay. So with this, we have studied about the conjugated lipids. In the next lecture, we will study one more example of conjugated lipid, that is lipoprotein, and then we will move forward to derived lipid. So that's all for now. Thank you.